Hello, welcome to the new session of Biomolecules NCERT revision. This is the fifth part of this chapter, and we are going to study about enzymes in this session. So, let us start with the definition of enzymes. So, all enzymes are protein. So, the chemical nature of all the enzymes that are proteins. There are some nucleic acid that behaves like an enzyme, okay, but their chemical nature is nucleic acids. So they are called as ribozymes. There are some nucleic acids that are behaving as an enzyme. So they are called as ribozymes. An enzyme, like any protein, has a primary structure. So the conformation, if you talk about the structure of a protein or the conformation of the protein, so they all have a primary structure. We have studied this in a great detail in our previous slide, that is the structure of protein. So there is a sequence of amino acid or a chain of amino acid in which they are connected. Each amino acids are connected with the succeeding one with the peptide bond, okay, and they form a primary structure in this way. So that is the amino acid sequence of the protein that is called as a primary structure. An enzyme, like any protein, has the secondary and tertiary structure as well. If we recall about the secondary structure, then they are having the alpha helix structure and as well as beta pleated sheet. So they can have only alpha helix or beta pleated sheet, or they can have both in one protein. Okay, so that is the secondary structure. Now the tertiary structure. Now this alpha helix and beta pleated sheet get fold in a proper way. And form a structural unit. Okay, that forms the structural unit. That is the tertiary structure. So all the enzymes are having primary structure, secondary structure, as well as the tertiary structure. At tertiary structure, the backbone of the protein chain folds upon itself. The chain crisscross itself, and hence many crevices or the pockets are made. Now this is very, very important. When the protein get folded to form the tertiary structure like this, okay, so there are some crevices you can see here. These are the crevices. Now what does this crevices means? These crevices or the pockets are having active sites. Now this is very important if we are studying about the enzymes. Enzymes are having active sites. At this active sites, the substrate get attached to the enzyme. A substrate comes and get attached to this active sites only, not at any other position, only at the active sites. So this active site is very important if we are studying about the enzyme structure. And this is formed by tertiary structure. So question can be asked that this active sites in a enzyme is formed by primary structure, secondary structure, tertiary structure, or quaternary structure. So the answer is tertiary structure. So active sites are formed by the folding of a polypeptide and forming crevices or pockets that contain active sites. An active site of an enzyme is a crevice or a pocket into which the substrate fits. So this is very important. The enzyme, through their active sites, catalyze the reaction at a high rate. So if they are having lots of active site, means the substrate get attached to it, and later on it catalyzes the reaction and the product is formed. Substrate get changes to a new product, and this enzyme increases the rate of reaction. It increases the rate of reaction. So enzyme catalysts differ from the inorganic catalyst in many ways. But one major difference need to mention here. What is that? Inorganic catalysts work efficiently at high temperatures and high pressure, while enzyme get damaged at high temperature, say above 40 degrees centigrade. This is very important, and this is the actual difference between the inorganic catalyst and the enzymes. Inorganic catalysts, they generally work at the high temperature and high pressure. Okay, they increase the rate of reaction at high temperature and high pressure. But what about the enzymes? At such an high temperature, enzymes are proteinaceous in nature. So protein get damaged above the 40 degrees centigrade. Okay, but there are some enzymes that are isolated from the organism that live in the hot vent and sulfur springs. These are the very adverse environment in which certain bacteria live. And these bacteria are having some enzymes and proteins that withstand at such a high temperature. They are stable and retain their catalytic power even at high temperatures up to 80 degrees centigrade and minus 90 degrees centigrade. Low as well as high both. Okay. Thermal stability is thus an important quality of such an enzyme isolated from the thermophilic enzymes. So these enzyme organisms are called as thermophilic organisms because they can withstand at such an adverse condition or such a high temperature. Okay, so in that organism, the enzyme are not destroyed. The enzyme are isolated. They have been isolated from those organisms. So the enzyme withstand at such a high temperature. But these are some exceptions. It doesn't occur in all the organism. Okay. Normally above 40 degrees centigrade, the enzyme get destroyed. Chemical reactions. To understand the enzyme, let us first understand the chemical reaction. So how the chemical reactions occur. Okay. Chemical compound undergo two types of changes. Physical change and the chemical change. So you all know that in physical change, no new substance is formed. No new substance is formed. So simply refers to change in shape without breaking of bonds. They change the shape, but there is no breaking of bonds. If there is a breaking of bond, then it definitely form a new compound, a new product. But here, new product doesn't form in the physical change. So this is a physical process. This is a physical process. Another physical process is the change in the state of matter. We all know that ice melts into water, and when water are heated, it becomes vapor. So here, the water can exist in three forms: solid, liquid, as well as gaseous form. Okay, so this is just a change of state. A new substance is not formed. So again, this is a physical process. So shape change, change of state of matter doesn't shows the chemical reaction. Now, then what is chemical reaction? The chemical compound undergo two types of changes. Again, here in the case of chemical change also, chemical reactions also, two types of changes occur. The physical change simply refers to the change in shape without breaking of bonds. So this is the physical process. We have already started that there is no breaking of bond in a physical change. So that is called as physical process. Another physical process is the change in the state of matter, that when ice melts to water, we have already covered this, that when ice melts into water or when water becomes to vapor, this is just a change of state. So this is also a physical process. However, when a bond are broken, a new bonds are formed during the transformation, this will called as a chemical reaction. So here barium hydroxide, okay, react with sulfuric acid, H2SO4. So barium sulfate, this sulfate get attached to barium and it forms barium sulfate and water is formed. So this is showing a chemical reaction because here the breaking of bond is involved. The breaking of bond is involved and a new product is formed. These are the reactant and these are the products. So here the chemical nature of these two substances are totally different from barium hydroxide and H2SO4. So this is a chemical reaction. Hydrolysis of starch into glucose is an organic chemical reaction. Rate of physical or the chemical process refers to the amount of product formed per unit time. This is very important, per unit time. In a given time, how much product is formed, that defines the rate of physical or a chemical process. So it is denoted by delta P. That is the amount of product formed in a unit time, that is denoted by delta T, and that gives you the rate of physical or a chemical process. So rate can also be called as velocity if the direction is specified. Rate of physical and chemical processes are influenced by the temperature among the other factors. So you can increase or decrease the rate of physical or a chemical process by changing the temperature or even the other factors. This we will cover in the next session, that what are the factors that governs the change of reactions. The general rule of the thumb is that rate doubles or decrease by half for every 10 degrees centigrade change in their either direction. So when you change 10 degrees centigrade of any reaction, that reaction rate get doubles or decreases. Either you increase or decrease depending upon the temperature, you are increasing or decreasing depending upon this. Catalyzed reaction proceed at rate vastly higher than that of uncatalyzed one. So what is the difference between the catalyzed reaction and uncatalyzed reaction? Reaction is going on, okay? Reaction is going on in the absence of catalyzed reaction, catalysts. But in the presence of catalysts, that reactions undergo a high rate. This is the difference. So when enzyme catalyzed reactions are observed, the rate which be vastly higher than that of some uncatalyzed reaction. So let us see this with the help of an example. Carbon dioxide H2O gives carbonic acid in the presence of carbonic anhydrase enzyme. Carbonic anhydrase. So in the absence of any enzyme, this reaction is very slow. This reaction occurs without the presence of carbonic anhydrase also, but that is very slow, with about 200 molecules of H2SO3 being formed in an hour. So how slow the rate of reaction here? You can see only 200 molecules will formed in an hour. Okay. So carbonic acid, only 200 molecules will form in an hour when the carbonic anhydrase enzyme is absent. However, by using the enzyme present within the cytoplasm, carbonic anhydrase, this enzyme, this reaction speed dramatically with about 600,000 molecules formed every second. Now you can see how high the rate of reaction is in the presence of carbonic anhydrase enzyme. Same reaction. Okay. The enzyme has accelerated the reaction rate by 10 million times. Here in this example, 10 million times the rate of reaction has been increased. So there are thousands of types of enzymes, each catalyzing unique chemical or the metabolic reaction. So there are number of reaction going on in our body or in any other organism that are catalyzed by certain enzymes. The multi-step chemical reaction, however, each step is catalyzed by the same enzyme complex or the different enzyme, is called as the metabolic pathway. So glucose is changed to 2-pyruvic acid. This is an example of glycolysis process. So if you remember, this is a 10-step reaction. This you will study in the respiration chapter. This is a 10-step reaction, and for each step there is a unique enzyme. So this glucose is a 6-carbon molecule compound that changes to 2 molecule of 2-carbon containing compound, 2-carbon containing compound, that is pyruvic acid. Glucose is a 6-carbon containing compound, and here 2 molecules of pyruvic acid is formed here. Okay. So it is actually a metabolic pathway in which glucose becomes pyruvic acid through the 10 different enzymes catalyzed by the metabolic reactions. So these are the catalyzed metabolic reaction in which the glucose is converted to pyruvic acid, and this occur in a 10-step reaction. So there are the different 10 reactions, and for each reaction there is a specific enzyme. These reaction is started in the process of glycolysis in the respiration process. At this stage, you should know that this is a very metabolic pathway with 1 or 2 additional reaction give rise to the variety of metabolic end products. So there are number of metabolic pathway, they are connected to each other, okay, and every reaction is catalyzed by some enzymes. In our skeletal muscles that undergo anaerobic condition, lactic acid is formed. So normally we are having what? Glycolysis process. But sometime when we do a vigorous exercise, the requirement of energy is so quick that it do not undergo the glycolysis process. Instead, glucose undergo the anaerobic conditions and form the lactic acid. So under the normal aerobic condition, pyruvic acid is formed. But in anaerobic condition, what happened? Lactic acid is formed. In yeast, during fermentation, the same pathway leads to the production of ethanol or alcohol. Hence, it is different conditions, different products are possible. How do enzymes bring about such high rates of chemical conversion? So till now discussion, what we have discussed now, we understand that the chemical reactions increase their rate in the presence of certain enzymes. Now, how this enzymes bring about such a high rate? Let us understand that. The chemical or the metabolic conversion refers to a reaction. So what is reaction? Any chemical change or the metabolic conversion refers to a reaction. So the chemical which is converted into a product. Now there is a chemical. Suppose that is named as A. It converted to a product P. Okay. So this chemical which is converted to product P is called as substrate. That is called as substrate. And hence, enzyme, that proteins with 3-dimensional structure including an active sites. Again, this active site is very important. So here comes an enzyme. Here comes an enzyme. This is an enzyme. This is an enzyme which are having lots and lots of active sites. Now this chemical which is in a substrate get attached to these active sites and it break down. Bonds are broken down and a new product is formed. So these active sites are the site at the enzyme at which substrate get attached to it. Okay, so that converts substrate into product. So this active sites are present where? Active sites are present at the enzymes, and these active sites are present at the three-dimensional structure, and three-dimensional structure is formed by tertiary conformation of the proteins. Symbolically, this can be depicted as substrate changes to product. Substrate changes to product. Substrate S has to bind the enzyme at its active sites. So there are different active sites that are present at the enzymes, and these enzymes at these active sites of the enzyme substrate get attached within a given clefts or pockets. So where are the active sites are present? Active sites are present at the clefts, crevices or the pockets. Okay, so the substrate has to diffuse towards the active site. Substrate has to move or come to the active site and get attached to it. And there is a certain, you can say, a stage where ES complex is formed. ES means enzymes and S means substrate. So ES, enzyme substrate complex is formed. Okay, and this complex formation is a transient phenomena. So this is not a result. The result will be product. But before the formation of product, there is a transient stage or a transient phenomena in which enzyme substrate complex is formed. Now, this enzyme substrate complex after a certain time get detached and a new product is formed. So here we can understand with this particular diagram. So formation of product with enzyme catalyzed reaction. So here you can see green color substrate is there. They are having some, you can say, clefts, and at the contrary of that cleft, again the enzyme is also having, this is an enzyme. This is an enzyme that is having active sites. So these are the active sites. Now this substrate come and get attached to these active sites. So you can see that it is something like a lock and key. Okay, lock and key type of things. So that is called as enzyme substrate complex, and this is a transient stage. This is the transient stage. So enzyme changes shape slightly as a substrate binds. So as the substrate binds to these active sites, enzyme started changing its shape. And due to the changing of this shape, bonds are broken down in the substrate. So here you can see enzyme products complex. Now the bonds are broken down here and the two products has been formed. So product leaving active sites. Once the product is formed, this active sites are not suitable to get attached to it because the enzyme has now changed its shape. So the products are now released out. In this way, the enzyme catalyzes the reaction. In this way, enzyme increases the rate of reactions. So during this state where the substrate is bound to the enzyme active site, a new structure of the substrate called transient state structure is formed. We have covered this. Very soon after the expected bond breaking. Now this is very important. One, in the transient stage, bond breaking takes place in the substrate and it is completed. The product is released from the active sites. When this bond breaking is completed, the product is released from the active sites. In other words, the structure of the substrate get transformed into the new structure, and that is called as product. The pathway of this transformation must go through the so-called transient state structure. So every enzyme catalyzed reaction undergo this transient stage, and in this transient stage what happens? Transformation. And how does the transformation occur? Transformation occur by the breaking of the bond in the substrate molecule. So there could be many more altered structural states between the stable substrate and the product. Implicit in this statement is the fact that all other intermediate structural states are unstable. So this is unstable, and that is why it breaks the bond. Stability is something related to energy status of the molecule or the structure. So why the product is formed? Because they want to get stable. Because when the enzyme is started changing its shape, the substrate is not having at the stable condition at that particular time. So what they do? They breaks the bond and a new product is formed. So this is the energy related things. So now how the enzyme catalyzed reactions are related to the energy, let us understand with the help of this graph. So when we look at this pictorially through a graph, it looks like something as in the graph. So this is we can see at x-axis progress of the reaction is there, and here we can see the potential energy. Okay. So now we can see here a substrate is here. Now let us having a two condition. Activation means the reaction taking place without enzyme. The reaction taking place without any enzyme, and the same reaction taking place with the help of enzyme. So what you see here, it requires lots and lots of energy to form the product when enzyme is not present. And when that same reaction takes place in the presence of enzyme, only small amount of energy is required to form the same product. So this is called as transient state, and this is the activation energy required without enzyme catalyzed reaction, and this is the activation energy. Now you can see how much energy is required. Okay, so the y-axis represent the potential energy content, and x-axis represent the progression of the structural transformation or the state through the transient state. The energy level difference between S and P. Substrate, here the energy is different, and here the product. Now you can see the energy is different. The P is at the lower level, and S, the reaction is an exothermic, means energy is released out and the product is formed. So one need to supply energy by heating in order to form the product. However, whether it is an exothermic or spontaneous reaction or an endothermic or energy requiring reaction. Okay, so here we can see that activation energy for the enzyme catalyzed reaction and activation energy without enzyme catalyzed reaction. So this particular stage is called as the transient state. So transient state is for this enzyme catalyzed reaction is less as compared to the reaction in which no enzyme is present. So S has to go through a much higher energy state or the transient state. The difference in the average energy content of the S from this transient state is called as the activation energy. So this transient state defines you the activation energy. So how much activation energy you required in the reaction in which no enzyme is present, and the reaction in which enzyme is present. So enzymes eventually bring down this energy barrier, making the transient S2P. So now what is the conclusion? This transient state is lower down. Less energy is required in case of enzyme catalyzed reaction. Okay, so this is all about the enzyme, how the enzyme react, how the enzyme catalyzed reaction work. So we will meet with one more session of enzyme. Okay, till then, thank you very much.